Yeah. So thank you very much. Oh, awesome. Thank you very much for this introduction. I am Alejandro Izquierdo López. I'm a postdoc in Northwest University in Xi'an, so I'm joining you from China. And on behalf of my co-authors, Dewang Jiao and Stephen Pates, I'm going to present you this small project we are working on called Exploring the Functional Difference between Early and Late Phanerozoic Arthropod Suspension Feeders. So, of course, the topic today is about suspension feeding. Suspension feeding is basically a lifestyle or a way to capture prey that involves like the animal basically trapping food from suspended particles, so trapping them from the water medium. So this type of lifestyle is present in multiple phyla across the animal kingdom, ranging from arthropods to brachiopods, sponges, or even whales. So all of these suspension feeders trap prey in different ways. For example, the most common one is using a filter. But for example, in the case of sponges, what they do is basically they use canals to basically do this type of suspension feeding. So overall, suspension feeders are very important for the ecosystems. They enable connection between different trophic chains, and they are able to recycle matter across the water column. So they have had a long importance throughout the history of animal evolution. So among all of them, what we want to focus are arthropods. So arthropods don't have a very high diversity in functional morphology because arthropods, most of them follow the same pattern. They have this type of appendages with setae or spines that they use to basically filter the prey from the water column. So you can see, for example, this image of a shrimp. So a priori, you would say that there's not much variation in how they do suspension feeding. However, if we look into detail, we can see more functional morphology variation in terms of the type of prey that they capture, their habitat, their movement, and so on. So basically what we want to do is basically quantify how these animals are doing suspension feeder and align them to different types of niches. So each niche will have different physiological requirements inside this umbrella of suspension feeding. So the main question we have as paleontologists is how has this functional diversity of suspension feeding changed between the in arthropod suspension feeders between the early Phanerozoic to today? In order to do that, we have to do two things. The first one is to obtain arthropod suspension feeders from the fossil record, and the second is to quantify this functional diversity to get a really good grasp about what was their exact niche. But while doing this project, we have the first problem, no? Which is preservation. In many cases, we can basically gather information about one animal, whether if it's an animal is a suspension feeder or not, based on things like biomechanics. However, what we really need is to find the filtering mesh. We need to find the filtering structure. This is the hard evidence we need. But this is, of course, very difficult to find in the fossil record. These are like structures that are feeble, that are small, so they're difficult to preserve. So the solution we found is, of course, taking advantage of the increasing examples of suspension feeding arthropods that are appearing across the fossil record, especially within a limited time frame. And this limited time frame is the Cambrian to the early Ordovician. These two eras together, it's not only the first origin of the animals and the start of the Phanerozoic, but also shows really, really good fossils of arthropods. So here are some of the examples of the fossils you can find during that time. For example, you find microarthropods. So these are basically millimeter-sized arthropods, which they're filter appendages that are around micrometer size. So that's the quality of the fossils we can have. We also have things like, for example, spines that obviously create meshes, like we see, for example, in this figure here, and other types of structures that evidently have this filtering function. All of these examples are part of animals, of types of arthropods that do not exist nowadays. So therefore, we can expect that there will be some differences between how arthropods were doing suspension feeding back then to how they do it now. So to compare them, we have this data set that combines both groups. And the main focus on the data set is prey size. So prey size we calculated basically by measuring the spacing between the filter meshes. So we basically measure across different fossils how big is the mesh, and using equations that have been published before, we can know what type of prey this mesh is able to capture, from micrometer size to micrometer to nanometer. Also, we have experimental evidence for arthropods that exist today. So there's multiple experiments that show how arthropods are filtering and what type of prey they can capture. So all of them together have prey size as our main focus. And from that, we can create different physiological variables. One of them is the relationship between their prey size and their body size, which we call prey to body size, which tells us about what type of energetic requirements does the animal have. Another thing is, of course, both maximum prey size and minimum prey size can themselves be informative. We may have two arthropods that have more or less the same prey to body size relationship, but their type of prey that they can capture is different. And finally, we have things like prey specificity. No, we want to know if an arthropod is focused on only one type of prey, or it can gather very different types of particles. So all in all, we have around 50 species combining fossil, extant, and some vertebrates for comparison. And basically, we do some type of transformations. And to summarize, we create PCAs that create this type of functional morphospaces. So here are some of the results that we have had so far. So one of the key results is that there is a big difference between what can arthropods capture at the minimum prey size and at the maximum prey size. So if you look at the different types of prey that they can capture and you look at the minimum, you have a very big variation. So arthropods can capture from the picometer to the mesozooplankton. No? So this is beyond like 200 millimeters. And basically, with maximum prey size is the opposite. So most of them are highly restricted to mesoplankton or microplankton. We also have some differences between the fossils and the extant taxa. So the fossils, for example, we have most of them only achieve preys that are very high in body size to the micro to the mesoplankton, while extant types can gather more different types of particles. This is the same, for example, with prey to body size. So if we look at the fossils here, we have that most of them have very low values of prey to body size relationship. So this means that fossils basically were only able to capture prey that were more or less similar to their own size. On the other hand, with broadness, we find that fossils are focused only on very few types of prey. So basically, the broadness is very narrow, is very low. So all in all, we have these very interesting changes between the extant and the extinct suspension feeders. But we also have to understand what type of groups have we lost since the Cambrian and the Ordovician. So one of the groups that we have found is what we call the Cambrian specialists. Most of them are, of course, from the Cambrian period, and this group different groups like early crustaceans, lobopodians, or radiodonts. And some of these arthropods have, for example, medium to high minimum prey size. They have low body to prey to body relationship, and they have a very low broadness. So that's why we call them specialists. So basically, they only focus on prey that are very similar to their own size, and also it's very limited broadness of prey. And the other group that we have found are these Ordovician giants. So what happens during the Ordovician is that some of these arthropods reach really, really high body sizes. Some of them were even like around 2 meters in length. So these Ordovician giants are a little bit different on their physiological requirements. So for example, you have a medium foot to body size relation and a little bit of medium type of broadness. So these are the two main groups that we have lost. And one of the ideas is these are these related, for example, in terms of niche to modern whales, for example. So sometimes in the fossil record, we call them the whales of the Ordovician. So is that the case? So in order to do that, we put some vertebrates to compare, and we have one PCA with the residuals and one without the residuals. So one has the influence of size, the other one does not. So without the influence of size, we have here on the right that it depends. So some of these Ordovician giants plot more or less closely to giant vertebrates like whale sharks, but other ones, like for example, actual whales, plot very differently. If we put the influence of body size, then of course, all of them plot very together. So that means that body size also influences a lot on whether we consider them to be on the same niche or not. But in any case, some of them were really close to manta rays, for example. So again, the difference between the two lost groups that we have had are, for example, the Cambrian specialists and the Ordovician giants. And we have that the Cambrian specialists have to medium to high maximum prey size, while the Ordovician ones have a high one. The foot to body size is low compared to medium, and broadness is low compared to medium. So to put everything into context, a little bit of the story we can see throughout the early Phanerozoic is this one. During the Cambrian, we have simple ecosystems, which very limited low plankton diversity and low plankton abundance. What we have here is suspension feeders that with very simple filters. So the filters of the Cambrian don't have subsections. So nowadays, if you look like a setae, you have that this setae has smaller setae, so they can capture more types of prey. Here, you only have one setae, so one like small like hair. So these simple filters, of course, can only capture one type of particle, so therefore they have low broadness, and usually it's around their body size or very close to their body size. So that means that some of these suspension feeders we could be actually comparing them to predators, or may need some types of other types of additional types of feeding that is not purely suspension feeding. And this is evinced by other adaptations and other speculations on how these animals how these animals live during the Cambrian. For example, this animal here has very large eyes. So previously we had been talking about whether this animal maybe was a predator, and now we know that was a suspension feeder, but maybe was also doing predatory behavior at a certain point. So afterwards, during the Ordovician, we have more complex ecosystems with high plankton diversity, and this allows for a higher type of size. And this also allows, with a higher size, you have higher broadness, so the animal can capture more types of particles. And also, of course, being larger means that there is a higher difference between prey size and body size. So this created this type of niche of very large animals with extremely specialized on suspension feeding. And what has happened since then is, of course, that they have gone extinct. What are the reasons? We don't really know. For Cambrian specialists, they were extinct in the Cambrian. We don't really know if they continued to the Ordovician because the fossil record for this type of animals is not as good as in the Cambrian. And for the Ordovician giants, we know that they continued through the Paleozoic, but they were reduced and eventually went extinct. And some of the causes could be competition, or basically, while having this whale type of niche was very special, and then for some reasons it didn't continue through the Paleozoic. So the conclusions is that if we evaluate suspension feeding in arthropods today, we have a wide variety related to their physiological parameters, and we can see, for example, a general rule of constraint at the minimum prey size. So very big arthropods cannot gather very small prey due to constraints due to energy. And of course, we have some differences between extant and extinct arthropods, including in things like broadness, or the type of prey, or the foot to body size relation. And finally, we have these two main groups that we have identified, that they are also slightly different between the Cambrian to the Ordovician. So with that, I will be finished with my talk, and I will thank you for your attention. And I would also like to thank the organizers of Evolution 2026 in a virtual format, because for many of us, it's extremely difficult to attend any conferences in person. So this is the only way that we have to share our research. So any questions are welcome. Thank you.
Nice, sir. Very good presentation. The platform is open for questions.
I have a quick question, if that's okay. Awesome. Yeah, I was wondering about the size of the mesh. You said like some of them can eat like pico and also micro, and that feels like a really big jump. Is this like because they have multiple different types of meshes, or is that because like the one mesh is very flexible? I guess I was kind of wondering.
Yeah, it's a little bit of both. So what happens is that first of all, arthropods can have different types of meshes on different appendages. So we may have meshes that are part of the mouth, so therefore like of the maxilla or the maxillula. So these are very thin, and then larger meshes that are part of the legs. Also, what happens is that the mesh basically has subsections. So you can imagine this as a kind of like a Christmas tree. No, you have like the main branch, and then you have smaller branches. So therefore, the animal can capture at the bigger size, can capture with the big trunk, and at the smaller size can capture with smaller branches. So this also creates this difference. And also sometimes these animals are very facultative, so sometimes they can change what type of prey they prefer depending on the prey availability or depending again on their physiological requirements at that point of time.
Oh, wow. Thank you so much. That's so cool.
Thank you, sir. Now we welcome our next speaker, Ido Filin, sir. Some new leads on coiling in gastropods. So you may start sharing your screen.
Do you see the slides now?
Yes, sir. Visible, sir.
Okay, so hello everyone. I'm Ido, and I'm speaking to you from Finland today. And I want to thank again the organizers for the opportunity to speak at Virtual Evolution. And I'm going to talk today about coiling in gastropods or coiled shell, theoretical morphology of coiled shells, which is a subject which I guess is familiar to many of you. So it is an old problem, and there has been many attempts to understand the great diversity of molluscan shells and gastropods in particular, and they go back many decades and many scientists. And there is and also very recent work, as you can see by this very incomplete list of names. And the reason for it is that it is a problem that it touches at the very core of evolutionary biology and biology in general, which is the interplay of function, construction, and history or development and phylogeny. And my goal in this talk is much more modest. I intend to convince you that the very familiar condition by Raup for whole overlap or for tight coiling, while it can explain covariation in coiling parameters, it is also explainable, or that covariation is explainable by a much simpler condition for the slopes of the conic spiral that defines those shells. So the allure or also the problems of studying coiled shells is exemplified by this quote from Monet. It is that it is a very simple morphology, and it is very highly integrated. That means that it's a very low dimensional morphological or morphospace, morphological space, or parameter space. And we can actually exemplify that by considering the center line of the shell. Its shape is completely defined by two parameters. For example, apical semi-angle and the slope of the downward slope of the shell, of the conic spiral that is at the center of the shell. So these two parameters completely define the shape of this centerline spiral of the shell, but because it's a very low dimensional morphospace, we can choose any other pairs of parameters. So it could be apical semi-angle and expansion rate, and you can see all the other examples here from the history of the subject. On top of these two parameters for the center line, we need the initial conditions, when to begin to grow, when to begin and when to stop, considering the growth of the shell, and some parameters for the generating curve, but the entire surface of coiled shells can be described by 5 or 6 parameters, depending on how complex situation you want to model. Now, because there are so many different ways to choose parameters, that means that also hole overlap and the condition of tight coiling can be described in different ways. So if you consider these two shells, one is tightly coiled on the right, on the left, open coiled. These two shells have the same apical semi-angle, like at the top, the same apical semi-angle beta. The open coiled clearly has a much faster expansion rate, so much higher gamma. But the same condition for open coiling or tight coiling can also be described by the lead angle, by the slope of this centerline spiral. So it actually gives a much simpler condition. It's just a constant. If you are below a constant, then you are tightly coiled. If you are above the constant, you are open coiled. And this is clearly, again, visible in these images. For the tightly coiled, the slope is much shallower. For the open coiled, it's steeper. And of course, this condition varies with the shape of the generating curve, and I consider later a certain variation of shapes. So we have alternative explanation, basically, from these relationships. We have alternative explanation for a hole overlap. There is a traditional due to Raup condition, which gives a covariation of apical semi-angle and expansion rate. And this may have some adaptive value in terms of mechanical strength of shells that are tightly coiled. But tight coiling can also be a side effect, and that is caused by a constant lead angle. So if lead angle is constant, given by this, then this constant times apical semi-angle gives us the expansion rate. And again, we get covariation of expansion rate and apical semi-angle. And it could be a mixture of the two explanations. So to examine how actually observed covariation relates to these theoretical curves and equations, I collected some data sets that cover different types of variation: ontogenetic, geographic, interspecific, temporal. And I examined how the scatter of these data sets relate to these theoretical relationships. And just to prepare you, in the following slides, the unshaded area and the solid black line are explanation based on constant lead angle. So lead angle is constant along the solid lines or the unshaded area, and the dashed lines describe the hole overlap condition. So let us first look at this data set from Nishita and colleagues. Regardless of whether we are looking at marine or freshwater or land species, there is most of the variation falls in this unshaded area defined by constant lead angles. And the linear trend, which is both the linear trend in the data and the scatter, nicely follows this straight line of either hole overlap or constant lead angles. Now, this covariation in the apical angle disappears when we are examining lead angle. So it does suggest that the axis of independent variation is actually lead angle and expansion rate and not apical angle and expansion rate. That if we want to build a morphospace of independent variation in each axis of this morphospace, then we need to choose the lead angle. This is the data of Newkirk and Doyle, which examined 5 populations, examined embryos and adults of 5 populations. These are population means. Again, there is a linear trend in the apical semi-angle view. The trend disappears when we are talking about, when we are looking at lead angle. It's even more visible when we zoom in. When we zoom in, we can see that even within the embryos and within the adults, there is this linear relationship, which largely disappears when we switch to lead angle. And there is some suggestion that embryos and adults are different, have some difference in lead angle as well. This is the data of Davoli and Russo, and this is like temporal variation. Again, within each epoch or age, there is linear variation between apical angle and expansion rate. These are again sample means. This trend mostly disappears in the lead angle, that at least when we compare this, when you compare the temporal variation, the different ages or epochs, there is basically no change in lead angle, except perhaps for adults from the Pliocene. And this is a much larger data set. These are individual specimens, not population means anymore. And we can see again linear trend within each group between apical and expansion rate and overall, of course. And this linear trend disappears both overall and within each group when we switch to the lead angle view. And we can better now understand what happens in the scutulata case, which has more or less the same expansion rate as the rest of the Tulospira, but has jumped up in lead angle comparable to the much higher spired Maori corpus. So what are the takeaways? First, tight coiling seems to be explained just as well by constant lead angles than by tight coiling for its own sake. And the combination of lead angle and expansion rate compared to the traditional apical angle versus expansion rate seems to be a better choice of a morphospace of independent variation. Now, because logarithmically coiled cornispiral shells are so highly integrated, as I referred to in the beginning of the talk, that means that there are many different ways to break this high integration. One way is suggested very much by the data, is that lead angle is fixed, but expansion rate changes, growth changes ontogenetically, geographically, and the apical angle is just dragging along as a passive consequence. Another way to change is, of course, for the lead angle to change itself. Now, in both cases, for example, if growth changes ontogenetically within an individual, so both expansion rate and apical angle would change within an individual.
Sorry to interrupt, sir. 1 more minute is left.
Yeah.
Thank you, sir.
It means that you'll get allometric shells, but the mechanism and the overall morphologies would be different. And the final takeaway is that there's lots of older data there, and it's worth a second look. So thank you.
Thank you, sir, for your presentation, sir. So what kind of phylogenetic corrections you applied, sir, for this? I mean, did you apply any phylogenetic corrections for those shells of?
Well, I didn't give any statistical analysis here. It's just like visual, but I did apply mixed effect models to study in the colonizal study because they give individual trajectories within each individual, ontogenic trajectories for each individual. I applied mixed effect models there, which can account for the effects of family and clades, separation clades. Yeah.
Thank you, sir. Thank you, sir. Now let us welcome our next speaker, Krista Koeller. How to measure the length of a snake's neck. Ma'am, you may start sharing the screen.
Is that good? Can you see it?
Yes, ma'am. Visible.
All right. Wonderful. All right. So as tetrapods, we have a body that is divided into different regions. This is mostly based in the vertebral column, but it also involves the limbs, and usually the divisions between the body regions occurs at the limbs. Body regions are distinct. You can see a difference between the trunk vertebrae or the torso vertebrae on the left and the neck vertebrae on the right. They're just very different shapes, and this has implications for function. The neck is the focus of my research. It serves a very important purpose in evolution. It is one of the factors that allowed us to go from the water to the land. It supports muscles that move and stabilize the head, as well as the limbs. It contains the hyoid apparatus and contains important passages between the head and the body, such as nerves, the trachea, the esophagus, blood vessels, etc. So it's an important region. My focus is on squamates, or the lizard group. They have distinct body regions like we do. And their neck, the neck trunk boundary, or the boundary between the neck region and the trunk region, is defined by the position of the shoulder girdle, specifically the first vertebra that has a rib that connects the shoulder vertebrae, or the shoulder girdle to the vertebrae. So this vertebra that connects to the shoulder girdle is the first trunk vertebra. And anything above that or in front of that is the neck. And usually in squamates, that's 8 vertebrae, pretty consistent across clades. However, there are instances where there is no shoulder girdle. And my question is, what do we do about those guys? One of the main groups, actually one of the only groups that doesn't have any arms or shoulder girdle is the snakes. Snakes are just weird lizards. They're derived lizards, and they have no forelimbs, no shoulder bones. And my question is ultimately how they became elongate. It's an evolutionary question. Their ancestors were just normal lizards, and over time they expanded. But they got really long. But how did they get long? Did they expand the neck region, or did they expand the trunk region, or did they expand both regions? And in order to answer that question, we need to figure out where the boundary between those regions is. And there are 2 potential markers that we can use as proxies since we don't have a shoulder girdle. One putative marker is these hypopophyses. These are on the bottoms of the vertebrae. They're just little bumps. And they serve for muscle attachments. Egg-eating snakes use them to slice eggs open. But they're really associated with the neck region in most lizards. However, in snakes, sometimes they go all the way to the cloaca. This particular specimen has 54 vertebrae with hypopophyses on them. And if we use hypopophyses as the neck trunk boundary marker, that means that sometimes snakes are all neck, and that there's a lot of variation within snakes for where the neck trunk boundary would be. Another putative marker is overall morphology. As we saw before, there's a big difference in morphology between trunk vertebrae and neck vertebrae. And luckily, we have a way to quantify this variation and compare it. I use geometric morphometrics, which is basically, to simplify, you simplify complex shapes, and then you can compare them and see how similar they are. In this case, we have a bunch of markers on different points that all fish specimens in this data set share, and those are plotted, and the shapes are compared. Basically, it's a way to mathematically describe complex shapes. And then what you do is you do a similarity analysis called a PCA, principal component analysis. And in this case, you can see the different, they cluster by similarity. So basically, all you need to know with these plots is the closer the dots are, the more similar the individuals are. Now, I'm using vertebrae, and the plots look a little bit different. They don't cluster so much. But this is just a similarity plot. This is not a graph. The X and Y axes do not matter. It's just about similarity. So basically, each vertebra is represented by one dot on this plot, and they just happen to line up because of, basically, I did a geometric morphometric analysis counting each vertebra in an individual as a specimen. And you can see they line up in corresponding order to the vertebral column, meaning that vertebra 3 is most similar to vertebra 4 is most similar to 3 and 5, etc. And I cut out 1 and 2 because those are weird vertebrae, and they would throw off the analysis. The atlas and axis are just different than the other vertebrae, so they're out. Now, I'm not the first person to think of doing this. Head and Polly in 2015 did that, and they found that snakes do indeed have distinct body regions. So it's not just a gradient. There are distinct regions that we are looking for. Now, of hypopophyses and the geometric morphometric determined boundary, how do we figure out which one corresponds to the neck trunk boundary? And I used legless lizards. So snakes are just a type of lizard, and they aren't the only lizards to go legless. There are actually over 30 lineages that have independently evolved in elongate limbless form or limb-reduced form. But most of them still have a shoulder girdle left. We can see in this figure, they're just kind of sticks. But that is technically a collarbone there and a shoulder blade, even though it doesn't look like it. But this is a way to tell where the shoulder girdle is on the body. So we can do an analysis using all 3 of those markers. We can look at a legless lizard, see where the shoulder girdle is, see where the hypopophyses end, and see where the geometric morphometric boundary ends. Or is, and then compare them and see which of the 2 putative snake markers would be a better fit. On my sample, I used all major limbless squamate clades, and I used CT data of limbed and limbless lizards. And I compared the shoulder girdle, the extent of the hypopophyses, and the shift in morphometric, the geometric morphometric signal. Now, pretty much all of my plots looked like this, where we have a sort of upside down V shape, where we have the neck vertebrae on one side of the angle and the trunk vertebrae on the other. And pretty much all of them looked like this. So we could tell that there was a distinct shift. And here is the summary of my results. This is a phylogeny of squamates, and I've starred pentadactyl. So like normal lizards are the ones with the stars, and the neck vertebrae, according to the geometric morphometric analysis, are yellow, and the geometric morphometric trunk vertebrae are in blue. The hypopophyses are marked by dots in the squares. And each vertebra is represented by a square. And then the position of the pectoral girdle, if it exists, is marked by an orange square. And it's a lot to take in. But there are some things that I want to highlight here. One is the clade Gymnophthalmidae. This is a group of lizards that has limb reduction, though not full limb loss. And their plot does not have a sharp angle. It actually has a curve, which means that there's sort of a gradient in morphology. It's not a distinct boundary, according to the geometric morphometric analysis. So that's interesting. I also want to highlight the pentadactyl specimens. So all of these pentadactyl specimens, they have their pectoral girdle in one place. They have their pectoral girdle is at the 9th vertebra, which means their neck vertebrae, there are 8 neck vertebrae in your typical lizard, which is what we expect. But if you look at the hypopophyses, so the dots, there's some variation, and it doesn't perfectly correspond with the position of the pectoral girdle. So that means it's maybe not a reliable indicator of where the neck is, even in limbed lizards. And looking at the geometric morphometric boundary, that also doesn't line up perfectly. It's a little bit closer of a match with the pectoral girdle or the shoulder girdle, but not perfect. So that's throwing a wrench in both of these hypotheses. Another interesting thing is looking at the position of the shoulder girdle. I've marked in orange here, the big orange rectangle is where the position of the shoulder girdle is in the pentadactyl specimens. And if you look to the right, towards the trunk, there's basically, in the non-starred specimens, there's very little there. It seems like most of the action is happening to the left, which means that basically no matter how you slice it, the neck trunk boundary is moving anteriorly with evolution. So it's over the course of elongation, the neck is actually getting relatively shorter in most of these species. There are some exceptions. Acanthus garapensis, right there in the middle. The geometric morphometric boundary has shifted to the right relative, or shifted posterior relative to the ancestral state. Same with the hypopophyses. But by and large, we have a shift closer to the head. I also want to highlight the snakes. I did only include 2 snakes in my analysis. This was actually mostly about legless lizards. But we can see that the geometric morphometric boundary is just a little bit farther anterior or closer to the head than the ancestral state. So they do have according to the geometric morphometric boundary, their necks have gotten a little bit shorter, which means most of the expansion has happened in the trunk, at least if that's our boundary marker. And the hypopophyses are all over the place. So that may not be the most reliable indicator. In conclusion, the neck generally decreases in length with body elongation. Hypopophyses are less constrained than other neck trunk boundary markers, so they may not be a good representation. And neither the geometric morphometric boundary or the hypophyseal boundary correspond perfectly with the limb position-based boundary. And this actually makes sense when we consider development. Body patterning is determined by a set of genes that basically tell the cells that they communicate with what they're going to be. And the limb tissue, or the future limb tissue, is actually patterned separately than the tissue that becomes the vertebrae. So it makes sense that they would be different and that they would be evolving separately. So my takeaways, the neck is not one discrete, consistently definable unit. It's different depending on what you're looking at. The neck trunk boundary markers are loosely associated, but functionally and developmentally distinct. And the neck trunk boundary evolves in similar ways across squamate groups. I am looking for PhD programs in the fall. So if you're interested in exploring the evolution of vertebrae and lizards, I'm I am interested. I'd like to acknowledge a bunch of people, and I think we have maybe 30 seconds for questions.
The podium is open for questions. Ma'am, any other group in which the neck is similarly contracting? Any other group other than
We don't
snakes?
know. So it seems like in lizards, that's the general pattern, but it has not been looked at in amphibians. So there are legless amphibians, a few groups, and there's about 25 fish species that have elongate bodies, but they don't have necks. But they do have some degree of regionalization. So that is another future direction that I would love to look into.
Yes, ma'am. So let's welcome our next speaker, Maya Enriquez. Ma'am, please please share your screen.
Great. Can everybody see that?
Yes, ma'am, visible.
Looks good. Okay, let's go ahead and get started. So today I will be talking about my thesis work or my dissertation work, determining the genetic mechanisms behind the repeated evolution of cave traits in the Mexican tetra. So, why do we care about parallel evolution? I know, probably heavy-handed to mention, but I always like to go back to the who cares, and you are all familiar with Darwin and how he said Landland's forms most beautiful and most wonderful have been and are being evolved. However, I also like this alternative. This is a book by Dr. George McGee at Rutgers University, and he wrote something called Convergent Form Evolution or Limited Forms Most Beautiful, with the idea that, yes, while we have a lot of diversity, there are constraints and there are potentially like these peaks or best ways of doing something, like a wing often has a certain kind of shape or things like that. And so, in the interest of convergent, talking about parallel, I'm just going to cross out, say we're going to be talking about parallel evolution today. Now I'm going to explain why it is that we're looking at the Mexican tetra or Astyanax mexicanus. So, we know that trait convergence is broadly found in cave organisms. Here I have four animals from very distant taxa, right? A cave fish, a cave salamander, a naked mole rat, and also a cave spider. And although these are from very different lineages, they are sharing the same traits, which are a lack of pigment and also either a lack of or highly reduced eyes. So in Astyanax mexicanus, we have both trait convergence and parallel evolution. So this is a species that has two very distinct forms. We have a surface and a cave form, and you can see that even though they're very different, they're actually the same species, and they can hybridize and produce fertile offspring. What's great about this population or this species is that there are several cave and surface populations. There are currently about 30 plus documented populations that live in kind of this central eastern portion of Mexico. And we know from previous phylogenetic reconstruction of these populations that Astyanax mexicanus cave fish form have evolved more than once. We know at least twice. There's a lineage about 200,000 years ago into two, we call lineage one and lineage two, and within those we have many different cave and surface populations that are currently extant. So, for my dissertation, I have three overarching questions. One, how is it that cave fish diverged from a surface ancestor? From these reconstructions, we know that it was a surface form, that's the ancestral form. Two, do different cave populations exhibit similar behavioral phenotypes or different behavioral phenotypes? And do the phenotypes across populations exhibit similar or divergent genetic architecture? Currently, in my dissertation work, I'm going to be focusing on this in my talk. I'm actually very close to looking at the genetic architecture, but for today, I'm going to be showing what my phenotypic analysis has brought. So, The approach that we are using is high throughput phenotyping of traits of interest to genetically map traits to the genome through a QTL analysis. And what might those traits of interest be? Well, I am a behaviorist at heart, and so I'm very interested in some unique behaviors that you might not be evident from looking at them. For one, cavefish exhibit significantly reduced sleep compared to surface forms. Here we have a surface and a Petron cave form, and you'll see the surface fish sleeps about 6 to 8 hours every 24 hours. And the cavefish sleeps is about maybe not even 2 hours. It could be less than that. We use movement as a proxy for sleep. And so here we are seeing that there is a super divergence in behavior. It's actually the only known organism, as far as I know, that has such a divergent sleep behavior that is not determined by things like seasonality changes or reproductive kind of breeding patterns. It is just that is baseline. So we're going to be looking at sleep. And we're also going to be looking at a structure called neuromass. Neuromass are the sensory pressure structures along the body that help sense water flow. In a surface fish, you can see that there are far fewer neuromass than there are in a cave fish. And this is shown across all cave populations. They just have like 2 to 3 times the number of these sensory structures. And given that we know that sleep appears to be there's some sort of sensory grading that happens during sleep. That's why you don't wake up when your alarm first goes off in the morning. We were interested in teasing this relationship apart. Additionally, previous work in the literature has shown that sleep and neuromass appear to be interrelated traits in Astyanax mexicanus. Here we're just showing an example of kind of a dye that's revealing the neuromass, and you can see in surface there's far fewer in this cranial area than in the Pachon cavefish. And then there was this really interesting and provocative correlation showing that it appears that the fewer neuromass you have, the more you sleep, or something like that. And so we wanted to tease this apart. For my work, I am focusing on 3 cavefish and 1 surface fish population. So as you can see, they're spread across lineage 1 and 2. We have in red our surface comparison that we're going to be using. And then in blue, I have the Molino population in lineage 1. And then I have the Pachon and Tinaja population in lineage 2. Okay, so in order to do a study like a QTL where we're mapping genes, we need to create variation between 2 different kind of phenotypes of interest. And so what we did is we actually did, as I mentioned before, we can produce fertile hybridized offspring. So here we crossed a parental cave fish from 3 parental cave fish, like the father, to a surface mother, and then created an F1 generation. And after that, we then did it once again in crossing in order to create an F2 generation that has a lot of trait variation, which is really important when you're doing a QTL. Not only did we do this once, but we did this actually 3 times with 3 different cave populations, all bred to the same surface grandmother, we'll say. So, and we created a lot of hybrids, up to 2,000 in one population. Sorry, 901 population. A total of 2,000 fish over these 3 different populations. And just for brevity, I'm going to be using these kind of acronyms in order to refer to these guys. This is what my phenotyping pipeline looked like. I'm interested in, like I said, sleep and neuromass. And one thing I did forget to mention at the beginning is cavefish are super resistant to starvation. So we decided to see if we might see a bit of plasticity in there as well. What we did is we did initial morphology collection. We let individuals acclimate for 5 days before recording for 24 hours using a behavior tracking software. They were both fed and unfed individuals over the 30 days, and then we did a final recording at the 30-day mark. If you're curious, this is what our arenas looked like. We had essentially these little fishy apartments that were completely separate from each other, and we were able to track using a behavior tracking software called Ido Vision, which would extract data like acceleration and movement, and below a certain threshold we called sleep, above a certain threshold we called awake. And then, in order to quantify the number of neuromass, we used a fluorescent-specific dye to image these. As you can see, it's gorgeous. I think it looks like a constellation. We focused on the craniofacial area just because it is highly associated with different cave-like behaviors. Okay, so for our trait characterization, we first want to determine and establish, though it's been done before, that surface fish do differ from cave fish. Were we able to find that? Thankfully, yes. So here I have our starting, we'll call them parental populations. We have our surface, and then we have our 3 cave populations. As you can see, surface sleeps around 10 hours on average versus a cave are way less. 5 at the most in Tinaja, then followed by Molino, and then Pachón. Then we have our hybrids, which we had way more individuals for. So those bars I'm showing are actually non-significance bars. The rest are completely significant differences between different populations. And as you can see, we have a ton of variation, which is very, very good for our QTL. That is exactly what we're looking for, is just a lot of variation. Okay, for our neuromass, same thing. Whew, thankfully, right? We have our surface fish, and what we did is we standardized the number of neuromass in a certain craniofacial area, and standardized it to standard length of the fish. So the number of neuromass in the area divided by the standard length. You can see surface fish have the fewest or the least standardized number of neuromass, whereas in our cave population, they have way more, 2 or 3 times, like I had said before. And then here in our hybrid individuals, we do see like a larger spread, although not the full spread necessarily. Like it's a little bit constrained. We were hoping for a bit more variation, but we are still seeing a lot of differences. And again, all of these individual pairwise are different except for these 2 cave surface hybrids here. Okay. Now, let's talk a little bit about plasticity. I don't think I gave this enough credence. It's really amazing how these cavefish are able to deal in these nutrient-limited environments, and we don't really know exactly how they do it. Like, do they tend to like sleep? Is it possible that sleep is rescuing some of that lack of nutrition? Is it possible that they move around even more? And so here I have some baseline data just for parentals for sleep and starvation. So we have on the left day 0, on the right day 30, and the little lines are just showing the reaction between the same individual over 2 different time points. And so here we did not really see any significant differences between sleep in the fed individuals between day 0 and day 30, except in Pachón, where it seemed like there's just a lot of variation at the beginning, and then it was very little sleep. And then here, for our individuals that were unfed, we do see a significant difference in sleep, where individuals are sleeping more on day 30 after starvation in our Molino and Tinaja cave population, not in our Pachón population, and interestingly enough, not in our surface population, which we were quite surprised about, although we do see a lot of variation, and there seems to be a lot of crisscrossing of kind of reactions between day 0 and day 30 that I'm hoping will be teased out by a little bit more data than I'm analyzing. Then in our hybrid population, we weren't really sure what to expect here, but we saw highly, highly significant difference where almost all hybrid individuals, on average, were sleeping way more on day 30 on starvation versus that baseline of day 0. Okay, and then the major piece is, do we see sleep and neuromass correlated? The whole point of my paper. And yes, we do. I put a little asterisk just because we aren't seeing any significant differences in our parental population, which we would hope to first establish, or significant correlations, I should say, but we do see it in 2 of our hybrid populations. Additionally, while in one population, so here we have parental Tinaja population, we see non-significant kind of as you get more neuromass, you sleep more, or sorry, yeah, more neuromass, you seem to sleep more. It's the same trend in the hybrids. However, in our Molino, we see a different trend where it seems to be actually going down in the hybrid individuals, which gives me some reason to believe that there's something happening here. Maybe it could be the way that the genes are kind of working out, and there could be some segregation happening as well with the parental individuals. So we will be teasing that a lot more out in our QTL. We have some ideas of why it is we might be seeing these things. It could be that these phenotypic correlations could be controlled by the same genetic mechanisms, like a single putative pleiotropic trait. It's one pet idea that the community has as to why cavefish were able to survive, is all these cave-like traits were inherited in a suite of traits very quickly in order to survive. It also could be that they're getting to the same phenotypes, but very different genetic means, right? They all have a different kind of flavor of caviness, and we still are going to be teasing that out with our future QTL. This is not real data, fake data, totally fake, but this is what I'm hoping to see at some point. We'll get some peaks in our data between some of the phenotypic kind of things we're teasing out in our data set, that we'll see some peaks correlation between the genome and these areas of interest. And this is coming soon, hopefully for the in-person conference. I have a bit more time, so I do want to also shamelessly advertise myself. Oh no, something went very wrong. And my PowerPoint is unstable, so I guess I will just finish by saying that I am on the market for a position as a postdoc. I had a fancy slide showing that I'm really interested in lots of things, including neuroethology, just where I started for my master's, behavior, and genetics. And so if anyone is interested in those things, please contact me. And yeah, I will stop there, given that my PowerPoint has decided to stop on me. But thank you for your time.
Thank you, ma'am, for the talk, for the very nice talk. The podium is open for the questions. Any questions? I'm not very much familiar with fishes and all. I mean, just like you can consider it as a layman question. But
Of
is it
course.
anything like cave fish have are in more nutrient-rich environment because their sleeping is also lesser, and also you can see that they are able to adjust without eyes. So are they in more nutrient-rich or food abundant environment?
No, sorry. I probably should have prefaced this more. The cave fish are actually in very nutrient poor environments. It's caves where you really don't have a lot. Like if you look at the structure, like the food pyramid in a cave system is not going to be nearly as rich as it is on the surface. They will sometimes get nutrients flooding in into the cave. Every once in a while through flooding events, that has been recorded, but otherwise it's pretty poor. And we think that they can survive starvation a lot more than 30 days. It's months it could be. So yeah, thank you. No, not, I'm happy to answer layman questions. Kinga, go
Yeah.
ahead. I see your hand is up.
Awesome. Yeah, it's kind of related. Oh, sorry, that's my alarm. But I was wondering because it is more nutrient poor, wouldn't you expect them to sleep more? But you kind of said that they slept like way less. I was wondering if that's like a genetic adaptation, or do you know why?
Yeah, we have lots of questions. So we don't know. There's been a lot of debate as to whether these traits, like at first it was like it's probably in a cave. There's no negative implication of not doing these things. And so it could be due to like drift, evolutionary drift happening, or it could be that it's adaptive, or it could be something in between. So yeah, you would expect when you don't have a lot of nutrients coming in, you'd want to like cope through other means. And so I found it really, yeah, the fact that they're not sleeping, some people have suggested it's because it's so nutrient limited that they're just spending the whole time foraging and trying to find food. They do exhibit something like hyperphagic behavior where like they will eat until they're so big and so bloated, like they will just continuously eat it. We call it hyperphagy. And yeah, I guess I just thought it was interesting too to see that the trend seemed to be as they're more starved, they sleep more. So no, I'm not sure, and I'm hoping to tease that out more. There are some really interesting candidate genes that we have found in other QTL studies that explain other traits like pigmentation and eye loss, for example, is one trait that seems to be tied together. But yeah, I have a lot more data to finish phenotyping. I also have, as a sneak peek, I have day 50, like we actually recorded not only day 0, but also day 15. So like the intermediary point of starvation. And we're curious to see what those curves might look like. But yeah, no, they are a fascinating organism to study. And I really love, yeah, I love these crazy cave critters. So
That's awesome. Thank you.
Any more questions? I guess we are done. I sincerely thank all the speakers for their valuable time. I thank everyone for attending the session. We got to learn a lot. Thank you to all speakers.